spring break, and I can imagine coming back to this weather from wherever is gonna be difficult. But earlier today we had a fantastic breakout, and I really enjoyed it. I was impressed, whoever was attending. I was like absolutely impressed by your level of performance, and of course our TA, she did an amazing job. So for those of you who are not or didn't have a chance to go, I really want to encourage you to go. You will learn so much. So everybody was great, thank you. So today we're gonna change the subject. In the past 2 lectures we talked a lot about the mitochondria. And one of the things that I wanted to emphasize and I wanted to kind of highlight was that the mitochondria were really necessary for the evolution of higher eukaryotic organisms. And the same is true for chromatin. So we would not be here if our ancestor eukaryotic cells wouldn't evolve the structure that is called chromatin. So today we'll talk a lot. Oh, I'm sorry. One of the screens is not showing the rating. Okay, sorry. Okay. So today we're gonna focus on the eukaryotic chromatin and genome organization. And this is a really nice image. This is taken by the electron microscope, and I really like those type of images because they show you the real thing. So we cannot see chromatin using the regular microscope, but we can do it with the powerful electron microscope. And this shows how the real chromatin looks like, how it looks like in the nucleus. So of course you cannot see a lot of details here, but by taking thousands of different pictures and manipulating the chromatin, researchers can learn a lot, and we can make these beautiful, colorful models. And right now we have a pretty good understanding how the eukaryotic chromatin structure looks like and how the genome in eukaryotic cells is organized. And so we'll focus on that today. And of course, I really want to encourage you to look at those learning objectives and study them. So for today's lecture, please focus on 3 main things. Study the levels of chromatin organization. We'll talk a lot about that in a few slides. Also, focus on the histone modifications and histone code, what it means, what it does, what it means for the cell in general, and how it's able to regulate the processes within the nucleus. And also we'll talk a lot about the chromatin remodeling and different mechanisms for chromatin remodeling. And we'll focus mainly on the ATP-dependent remodelers, so using ATP energy, the ATP energy that mitochondria are producing, right? So this is a very important mechanism for chromatin structure regulation. And also we'll talk about histone acetylation. So I want to start by asking a very simple question. How long is our DNA? And I want to be specific. So we take one cell from our, let's say, skin, and we have 46 chromosomes. We take all of those chromosomes, purify them, unwind them, and organize them in the linear fashion. So we have this stretch. So how long this stretch is going to be? How long it is in human? 6. Somebody said 6. Yes, precisely. It's you're yeah, great answer. 6 feet. That's how long. I thought that you will have more trouble, but you're great. You're excellent. So the DNA inside each of our cells is longer than we are, typically, because if you're higher than 6 feet, then maybe that's not the case. But what is really intriguing is that such a long piece of DNA is able to pack down into a tiny space of our nucleus, and the nucleus is only 5 micrometers. So this is a compartment within our cells that we cannot even see. So this is quite intriguing. But in our body, in our whole body, we have 50 trillion cells. So now, for those of you who are good in math, how long the DNA is in our entire body? So somebody in the past did this calculation for you. So it's gonna be enough to go from the Earth to the Sun, so back and forth, 300 times, which is incredible. So that's the incredible length of the DNA that packs in our body, in average human body. So you may think, how is this even possible? Is it possible? And of course, there is a storage problem. So how does our DNA, genomic DNA, is able to fit into the small 5 micrometers compartment? And our ancestor eukaryotic cells already figured it out. So the question is, how do they do? What has to happen? Packaging, that's the keyword. That's the keyword for this entire lecture. So DNA must be highly folded and tightly packaged. So here we have a very general schematic overview how DNA is packaged into a eukaryotic chromatin. So this is showing a eukaryotic cell nucleus, which looks like a bowl full of spaghetti noodles, and you may see it as highly disorganized DNA, but in fact it's not. So here at the very basic level of organization, we have DNA that is packed, so it's wrapping around those circles, and we'll talk about the details of this process in a little bit. But at this level, so this is very basic level of chromatin organization, and those are nucleosomes. So those individual units are called nucleosomes. So DNA wraps around the histone octamer, so those are histone proteins, and so they form this basic structural unit of chromatin called nucleosome. And nucleosome then is folded with additional proteins, so it packs down and forms higher order chromatin structures. And then when DNA is very, very highly tightly folded, it forms a chromosomes, and they occupy specific territories within the cell nucleus. So they are not randomly placed in the nucleus. They have a specific place and structure. So why DNA is packaged into the nucleus in eukaryotes? So we know, for example, by contrast with prokaryotic organisms, prokaryotes don't have nucleus, and they have very small, simple genome. So why is it important to organize and package the eukaryotic genome and have it enclosed within the special compartment in eukaryotes? Why this is important? Any ideas? So do we need to protect our DNA from damage, from breakage? Yes, that's the key point. Those tiny little microbes, they have a very short lifespan. They can live like bacteria. Some bacteria can live only 20 minutes. We, like humans, want to live long, like 100 years perhaps. So we want to make sure that the DNA in our cells stays intact and protected. So this is the way when cells package the DNA and highly organize it, put it into the nucleus. This is a very good environment for protection of our genomic DNA. Also to keep it structured, to keep and maintain specific structure so it's not all tangled and disorganized. And also, of course, to separate those DNA templated processes such as DNA replication or RNA synthesis, transcription. So those processes are separated from the cytoplasm. So those are 3 key points why DNA is packaged and highly organized in eukaryotic cells. So this is, again, very simple model showing a chromatin structure. And chromatin, by the definition, is the DNA associated with proteins. And we can distinguish two main categories of proteins. Those are histone proteins. So you can see here histone proteins that are the key components of the nucleosomes, shown here in yellow. And we have a huge variety of additional non-histone proteins, and those are also very important. They play major roles in the organization of the chromatin, and also they regulate different processes such as DNA replication, transcription, or DNA repair. So those could be important transcription factors, DNA polymerases, and huge amount of other accessory proteins. And chromatin structure in our cells is not static, is highly dynamic, and it changes. During the cell cycle, there are dramatic changes in the chromatin. And we'll talk a little bit about that. I'm not going to talk about cell cycle, but I will try to, when we'll talk about different levels of chromatin organization, we'll kind of distinguish where these profound changes happen. So now let's focus on the nucleosome. So this is the basic structural unit of the chromatin, shown here. And these days we know a lot about the structure. So we know the crystal structure of the nucleosome, which is shown here. It's a beautiful structure, very regular. So when you look at it, it looks like a beads on the string, and that's also the term that a lot of scientists are using, because it really looks like a beads on the string. So this is the DNA wrapped around the histone octamer proteins. So this level of organization, DNA is compacted six-fold, and the nucleosome is comprised of the core of 8 histone proteins. So here we can see the core, or here in orange, and the DNA that is wrapped around. So there's about 147 base pair of the DNA that wraps around the core. And then there is this linker region, which is between nucleosomes, and it's about 20 to 60 base pair long. So it's very short. And at this point, I wanted to mention that histone proteins are among the highly conserved proteins. So they are highly conserved in all eukaryotic organisms, from tiny yeast cells to humans. So they are very conserved. They didn't change because they are important for this fundamental structure of the chromatin. So from the mechanistic point of view, why would DNA wrap itself around the histone core? So you may remember from your previous courses from the genetics or biochemistry, what would you say, like from the mechanistic point of view, why DNA wraps around the histone core? What would have to happen in order for this interaction and the wrapping? Would you repeat that? Yes. So there is a lot of interactions. So for the electrostatic interactions, what kind of interactions would be involved? So DNA, does it have a charge? Yes. It has a net charge. Is it positive or negative? Negative, yes. And what we know that those histone proteins are positively charged. So there is an electrostatic interaction between the negatively charged DNA and positively charged histone. And this is the main force that drives this wrapping phenomenon. And of course, it's energetically favorable. So something that has a negative charge attracts to the positive charge, and this is energetically favorable. So there are about 142 hydrogen bonds that form also between the DNA and the histone core of each nucleosome. And there are also hydrophobic interactions and salt linkages. So there are a variety of different interactions between the core histones, which are rich in lysine and arginine residues, which have basic side chains and positive charge. So, and this interaction between the DNA and the histone core is very tight, because all of those hydrogen bonds, even though they are weak bonds, but if you have so many of them, the final outcome is that this DNA is tightly wrapped around the histone core. So it doesn't fall off very easily. And histone proteins, they are small, positively charged, high in arginine and lysine, and we can distinguish so-called core histones, H2A, H2B, H3, and H4, and also there is a linker histone, H1. So this histone is going to bind in the linker region. And here in this panel A, we can see the structure of the histones. So what we can distinguish here is the histone fold, which is very organized region of those histone proteins, and a very specific they have very specific N-terminal tails, which have different length. And the important thing is that those tails are not structured or organized, so they are loosely folded. So this middle part is organized, so it's gonna fold like form a alpha helices or beta sheets in the protein folding, and those N-terminal tails will kind of like stick around, and they will be more like unstructured. So how do nucleosomes assemble? This is a very interesting and fascinating process because it can happen spontaneously. It doesn't require ATP energy. We said that it's energetically favorable to wrap DNA around the histone octamer. And this figure shows how it happens. So first of all, we have a histone H4 and H3. They come together and they form a H3-H4 dimer. And then we have another dimer, and two dimers come together. They form H3-H4 tetramer. And at this point, so you can see this tetramer, very nice, which is having here this central core, nicely organized, and those N-terminal tails sticking out. So at this stage, DNA is able to interact with this H3-H4 tetramer, and it's able to wrap itself around. But at this point, the nucleosome structure is not complete, because it needs the H2A and H2B dimer. So H2A, H2B come together, interact together, form a dimer, and two dimers are needed to build this beautiful nucleosome structure. So here we have a complete nucleosome which contains a histone octamer, and those N-terminal tails are sticking around pointing in different directions. And I said that this process is quite fascinating because it can happen spontaneously. So you can actually do it in the test tube. So you can purify the histone proteins, express in different systems, and purify those recombinant histone proteins. And you can take a piece of naked DNA, you can put it together to the test tube, and you don't need anything else, no enzymes, no ATP energy, and spontaneously DNA will organize, will start, wrapping itself around those histone octamer, and will organize, will create this simple chromatin. And this is a very powerful tool that researchers are using to study chromatin. So we can study those processes in vitro, so in the test tube. So we can look and modify this piece of artificial chromatin. We can put different modification on the histone tails and check how this structure is changing. We can use the electron microscope to look at this. So here you can see this very beautiful beads on the string organization, which looks like here. So we have individual nucleosomes nicely ordered in a linear fashion. So about histone tails, I've already said that. And we'll focus later on the modifications of the histone tails. So histone tails are highly modified by different chemical groups. So here we have a really nice example showing different histones, H2A, H2B, H3, and H4. And those are the tails. And they can be modified by different groups. So we can see methylation, phosphorylation, acetylation, ubiquitination, and modifications are put at specific places, at specific amino acids, and these modifications, the composition or the, how to say that? So different modifications create so-called a histone code. So they are not meaningless. They are encoding the specific information. And so, for example, methylation is often associated with gene silencing. And we are currently studying those modifications and trying to solve what are the different messages encoded by different modifications. And some of those tails can be really, really heavily modified. Like, for example, histone H3. It can have all kinds of modifications placed. So a lot of amino acids here are modified. Some can have like multiple modifications, like D3 methyl groups at the same amino acid. So those are highly modified histone tails. They are unstructured and they carry a specific message for the cell, and they play important roles. So right now, I want to talk about the chromatin structure. So this is a really nice panel showing different levels of chromatin organization. So as we go from this very basic level, which is naked DNA, double helix, 2 nanometer in width. And we've talked about this. This is the next level of organization, bits on the string or nucleosomes. And here, so those images are showing you the microscopic view. So when we look under the microscope, the electron microscope, and we see naked DNA, we just see the solid line. We can see these beautiful strings, bits on the string structure as well. And when we go higher, when those nucleosomes are folded into higher order level, so this is a 30 nanometer fiber. And it looks under the microscope like a solid fiber. So the resolution at this point is kind of lower. We don't see those really nice distinct nucleosomes because the structure is much more folded and it has a higher electron density. So the next level of organization is the loop. So the 30 nanometer fiber is looping, it's forming those large loops, and we can see them under the microscope as well. And finally, loops are folded into the higher orders. And finally, at the end, we have mitotic chromosomes, which look under the microscope like this. So we have telomeres and centromeres, and so this is a typical mammalian chromosome, highly so that the DNA is highly compacted. And this whole region is dark, is this electron density very high. So when we talk about chromatin, we often like to distinguish two main fractions. So we talk about heterochromatin and euchromatin. Are you familiar with those terms? Is there a difference between heterochromatin and euchromatin? So on this figure, what level of chromatin organization would you classify as euchromatin? For example, beads on the string. Would you say it's a euchromatin or heterochromatin? Yes, it's euchromatin. So euchromatin by definition is more open chromatin, more accessible, and typically within euchromatin you have gene expression. So this form of chromatin is open, accessible to DNA transcription, to DNA replication. Heterochromatin is more closed, inaccessible, typically silenced part. So here on this figure, we could say that beads on the string represent a euchromatin fraction, and also chromatin fiber can be also classified as euchromatin. Versus here, those loops, domains, and of course the mitotic chromosomes represent a fraction of heterochromatin. And here's the view of the chromosome with the centromere and the telomeres. So depending on the cell cycle stage, chromosome can kind of unwrap itself from this tightly folded conformation, but some fraction of the chromatin will stay very compacted all the time, and this is typical for the centromere chromatin and also the chromatin that is found on the ends of the chromosomes. Those are the telomeres. So here on the ends we always find very silent, inaccessible heterochromatin. And here we have another view of the eukaryotic cell. So this is an electron micrograph. Here we have a nucleus and two fractions, euchromatin and heterochromatin. So euchromatin is shown here as a lighter region. It's not so electron dense. And heterochromatin is shown here as those darker patches or regions. And what's specific in normal cells? Heterochromatin is found in specific locations. So when you look at the nucleus of the normal cell, you find the heterochromatin fraction on the outside of the nucleus, on the nuclear periphery. There is a heterochromatin, and there are also regions found inside. And typically around the nucleolus, you will find a fraction of the heterochromatin. And euchromatin, this transcriptionally active chromatin, is found in the center, is localized typically more like in the center of the nucleus. And of course, when you zoom in on the euchromatin, you may find the beads on the string structure. So this chromatin would be active, you would have an active gene expression going on. Versus here, there is no gene expression. This whole region is very tightly packaged and silenced. So beads on the string, we kind of talk about that. Yes, linker region. We didn't talk about histone H1. So histone H1 is important when we have the beads on the string structure, and histone H1 helps with the further packaging of the bits of this on the string, so helps tighten DNA where it binds. So here we have a histone H1 shown here in green. So it binds here at the bottom of the linker region, and it helps kind of bind this. So this is the entry point of the DNA. DNA wraps around and exits. So it kind of like binds at the bottom. Here we have a histone H1. So it's not part of the core. It's stabilizing the entire nucleosome structure more like at the bottom. And here we have a electron micrograph of the chromatin bits on the string without H1. So they are more like disorganized structure. When we have H1, they start forming like this 30 nanometer fiber structure. So very nicely organized structure. So the next level of organization is 30 nanometer fiber. It compacts DNA 40-fold, and histone tails may also be involved in those interactions, and they may help stabilize the entire fiber. And also the core histones, H2A and H4, may also cooperate to form this 30 nanometer fiber. And this is a different view. So 30 nanometer fiber can be organized under two models. There is a zigzag and solenoid model, and depending on how the nucleosomes are packaged, there is a difference between the zigzag and solenoid. And here's a different view. So maybe this will help you distinguish between the solenoid 30 nanometer fiber and the zigzag. So in the solenoid, when you take the cross-section through the solenoid fiber, you will see the kind of like an empty space here. So this is how those nucleosomes are organized. Versus in the zigzag, usually there is no empty space. So they are very tightly packaged and tightly folded. And DNA loops. So those 30 nanometer fiber, they form a higher level of organization loops. And those loops, so here we can see the loops, and they are called loop domains. They attach to the nuclear scaffold. So this green thing is the proteins forming a chromosome scaffold. So they are attached in order to maintain those loop domain structure. And here, for example, we have a domain where it's kind of like unwrapped and unfolded a little bit from the entire thing. So here we have a situation where we have a high level of expression of genes in the loop. So this loop would like relax and would form euchromatin. And in this region, you could have a gene expression. And of course, for this to happen, we would need we would have to remodel the chromatin structure here. So we would have to have additional proteins, additional complexes, so-called chromatin remodelers, to come here to those domains and unpack or release the chromatin from the very tight, compacted structure and form more open structure. Now we're going to have a short activity. So I encourage you to work with your partner in groups of 2 or 3 if you like. And so this is the activity. So this is the image, this is the electron micrograph image of the chromatin or the human chromosome that was treated with high salt, like a table salt, sodium chloride. So when you look at this image, try to discuss with your partner first, what do you see? What do you see here? I hope the resolution is good enough, but we can always speculate what we see. And so yeah, try to kind of ask or discuss with your partner what you see and why would high salt have this effect. So how about if we take like 2-3 minutes? All right, you ready? Who has an idea? Anybody? And you can guess. It's totally open for interpretation. So I really encourage, creativity. So first, what do you see on the picture? Euchromatin. Where do you see euchromatin? Here. And I can totally understand this is not the wrong answer, and I can totally understand how you could easily interpret this as euchromatin. And so why would you see like more open chromatin here? So this would be like euchromatin, or even like a naked DNA. Because I remember I told you when we look under, and this is the electron micrograph. And at this resolution, this is naked DNA. And all of this grain in the background, what would this be? So if this is naked DNA, what would this be? Yes, those are the histone proteins. What do we have here at the bottom? Yes, this is the fraction of the heterochromatin, or the rest of the chromosome that was treated with the sodium chloride. And now, why sodium chloride would do something like this to our mitotic chromosome? So what happened? So what happened here? So we've started with the fully compacted mitotic chromosome. Let me show you here. So we take this chromosome and treat it with sodium chloride. And sodium chloride would do something to this structure and convert it to this. So what happened? So based on what we know so far, the electrostatic interactions, yes?
Well, basically talking about how the histone and DNA interact with the positive part of the histone and the negative part of the DNA. So you're throwing NaCl is a ionic compound, so the first part you have a positive sodium ion that can interact with the negative part of the DNA, which will disrupt the interaction between the DNA and the histone to the first part, and you have that DNA.
Exactly. So sodium chloride, and you probably know what happened, so I don't need to explain. So pretty much sodium chloride was able to disassemble this compacted chromatin into a naked DNA, and so the histones were displaced from the chromatin, so we end up with the naked DNA and some fraction of the chromatin that was left. So we can, this is an important tool, sodium chloride or other salts, because researchers are using this tool in order to study chromatin and disassemble chromatin in vitro when they do experiments with chromatin. And for example, here we have a really nice diagram showing what can we do in vitro in the test tube to disassemble the chromatin. So we can use salt, and we can also use enzymes, nucleases. And there are special nucleases isolated from the bacteria, and the enzyme is called micrococcal nuclease. And this nuclease is able to specifically act on chromatin. And what it does, it binds to the linker region, and it's able to degrade the linker region. So the micrococcal nuclease acting on the chromatin will release the nucleosomes. So we can kind of like separate the individual nucleosomal units from the chromatin. And then we can treat with the high salt, and then we'll get a further separation of the histone core and the DNA. And we can also dissociate the histone core onto individual histones. And now we have a short movie kind of summarizing of what we've learned so far.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
So in the cell that is not rapidly dividing, our chromatin is not condensed in the chromosomes. And we talk about the interface state chromatin, and at this stage we can distinguish chromatin fibers, perhaps bits on the string, but not the tightly condensed chromosomes. So now we'll move on and talk about regulation of the chromatin structure, and we'll focus on mechanisms of chromatin remodeling. So if we talk about this, solving one problem where, long stretch of DNA has to be tightly packaged, compacted into the cell. But right now, we've solved this problem. We organize the DNA into a highly compacted, packaged chromatin. But at the same time, we create another problem. So what is this another problem? So in order for this tightly packaged DNA to function, what it needs to happen. So this DNA in the context of chromatin is highly inaccessible, right? So other proteins that need to gain access to specific target sequences, for example, to express specific genes, are not allowed. So what it needs to happen in order to allow for the gene transcription? Yes, so we talk about chromatin remodeling. So chromatin remodeling is necessary in order to facilitate DNA accessibility for those different DNA templated processes in order for this DNA to function in context of the cell. So chromatin needs to be dynamically reorganized or remodeled, and I just said to facilitate direct interaction between regulatory proteins and target DNA sequences during the DNA replication, transcription, and repair. So even though our chromatin is highly organized, it's not static. It's at the same time highly dynamic. So now we're going to talk about how cells, what are the mechanisms that are involved in active chromatin remodeling. And one of the mechanism is based on using the ATP energy. And we have a nucleosome remodeling complexes. And those are huge proteins, huge complexes composed of different subunits. And they have this central ATPase subunit, which is able to use ATP energy, and it can bind to entire histone octamer. And using the ATP energy, it can perform, it can directly interact, or it can alter the interaction between the DNA and histone octamer. And it can remodel the chromatin, so it can result in the nucleosome sliding, ejection, or exchange. We have a better figure to explain this process. So this is a nice figure showing our stretch of the chromatin. And for example, here, when we focus on this red piece, so let's say this is a very important regulatory element. And this regulatory element is important for the expression of the gene that is important for cell survival. And in this conformation, this gene, this regulatory element, is not easily accessible for DNA binding protein because it's wrapped around the histone octamer, and this protein, DNA binding protein, may not have a easy access. So what's going to happen? This ATP-dependent remodeler is going to be recruited to this place. It will bind to this octamer here, and it can use ATP energy to alter the interactions between the DNA and the histone octamer, and this can result in different chromatin remodeling outcomes. So in this scenario, the entire octamer can be ejected from this place. So now this red piece of DNA is available, so the protein can bind, and this can initiate expression of our gene. A different scenario can result, here at the top we had a repositioning. Here we have a ejection, so this piece is available for the DNA binding protein. And in a different scenario, we can have just a simple unwrapping of the DNA of the histone octamer surface. And now with this form, this red piece is available or visible to DNA binding proteins. And we can also have a different forms of chromatin remodeling involving dimer exchange or dimer ejection. So all of those activities are performed by specific group of enzymes called ATP-dependent chromatin remodelers, and they rely on ATP energy to perform all of those functions. And here we have an example of ATP-dependent chromatin remodeler, and this gives you a better idea of the scale, how large this protein complex is. So it's large enough to wrap around the entire histone octamer. So here we have a histone octamer kind of sitting in the cavity of this huge complex. And using ATP energy, this complex can alter the interaction and perform different chromatin remodeling. So we've talked about the experimental chromatin decondensation using salt, or we can use also different enzymes to kind of release the 30 nanometer fiber to unpackaged and kind of release the 30 nanometer fiber to the decondensed chromatin bits on the string. And in cells have a different mechanisms to also unpackage the tightly folded chromatin. But cells, of course, don't use salt to do it. They have a specific enzymes. And there are two very important groups of enzymes that are able to reorganize chromatin. We have a histone acetyltransferases, or HATs, and those enzymes are able to put acetyl groups on histone tails. So this is the modification that will convert a condensed chromatin to more open decondensed chromatin. So if this is a heterochromatin silenced, this histone acetyltransferase would acetylate the chromatin and allow for the opening of the chromatin structure and converting the condensed chromatin to euchromatin. And of course, this process is dynamic and balanced. So we have enzymes that are opening the chromatin, and we also have enzymes that are closing the chromatin. So on the other side, we have a histone deacetylases, or HDACs, and they remove acetyl groups from histones, and they allow for regulated condensation of the chromatin. And this illustration shows a chromatin accessibility. So this panel here shows a normal histones, and I've talked about the chromatin fractions, euchromatin and heterochromatin. That heterochromatin is typically located at the nuclear periphery. So here this fraction is the heterochromatin. And also here around the nucleolus, and this fraction is more open, accessible chromatin. And those blue balls represent different macromolecules. So of course, accessibility within chromatin here is only, so we have two compartments. So in the heterochromatin has a very limited accessibility, and only small micromolecules can access this compartment. And large macromolecules are only present in this more open euchromatin. And what's going to happen when we have a hyperacetylation of chromatin? So here we don't have those two fractions. So entire chromatin was converted, so entire heterochromatin was converted to more open euchromatin structure. So right now, all of those micromolecules have a even access virtually to any region within this chromatin. And we can kind of transition between this state and this state using a trichostatin A, which is a selective inhibitor of the HDAC enzymes, of the histone deacetylase enzymes. And when we treat cells with this inhibitor, what it does, it's going to permanently deactivate histone deacetylases. So we're going to end up with hyperacetylated chromatin. And hyperacetylated chromatin, it's going to be more open, more accessible. So when we have, for example, a gene that was not expressed because it was located perhaps in this fraction, in the heterochromatin fraction, now we treat the cells with the trichostatin A. This may allow for the expression of the gene that was located in the heterochromatin. So, and this is again really nice tool, trichostatin A, to manipulate chromatin in research and to play with different conditions to change or manipulate chromatin structure and see how that affects differential gene expression in different conditions. And at this point, I also wanted to show you a very interesting thing. In embryonic stem cells, we'll have a separate lecture on embryonic stem cells. In embryonic stem cells, there is this unique configuration of chromatin. It appears that they don't have any heterochromatin. So it looks like all of the chromatin in the embryonic stem cells, it's open and accessible. And during the differentiation, when the stem cells is differentiating to different cells, for example, neuronal cells or muscle cells, so during the differentiation, we have formation of heterochromatin. So different regions will now become the fraction where heterochromatin forms here. So in the differentiated cells, we have fractions of heterochromatin, which will form more like on the nuclear periphery and also around the nucleolus. But in the embryonic stem cells, there is no chromatin, no heterochromatin. And now we're gonna focus on the chromatin function and also a global structure of chromosomes. So I've showed you this. I've showed you that histone tails can be modified and that this modifications is like writing a specific message or writing a specific code. So there is a hidden meaning in all of this. We don't exactly know what it all means, but a lot of labs are trying to figure out. So, and we know a little bit. So we know, for example, that a trimethyl of lysine 9, K9, trimethylation, is the signal for heterochromatin formation and gene silencing. And those two modifications, K4 methylation and K9 acetylation, so combination of two different modifications, is the signal for the cell or for this particular region in the nucleus for the gene expression. So this can recruit a DNA, it can facilitate, it can allow gene expression. And for example, this modification is trimethyl, so we have 3 methyl groups placed on the lysine K27, and this encodes a message for gene silencing. So when we have this modification around the particular gene, this sends a message for silencing of this region and silencing of the gene expression. So writers, readers, and erasers in chromatin. And I really like this figure because it kind of like puts everything together. And so we've talked about the histone code, components of the histone code. We talk about the modifications, and that those modifications are placed on the histone tails at specific places, and those modifications are placed by specific enzymes. So methylation, metal transferases are responsible for putting the metal groups, acetyltransferases for acetyl groups, ubiquitin ligases and kinases for phosphate groups. So this process is called writing. So those enzymes will write the histone code. They put specific modifications at specific places. And of course, to balance this process, we have to have enzymes that remove those modifications from the histone tails. And those are called erasers. So for methylation, we have demethylases, or acetylation deacetylases, deubiquitinases, and phosphatases for the phosphate groups. And when we have different combinations of those modifications on the histone tails, this is a message for a different group of enzymes called readers. So we have those complexes that recognize specific combinations of modifications, and they can be recruited to this particular place, and they can read the message, and they can then trigger some downstream events that will then translate to specific, it can change specific processes within cell. So we have metal readers. So those metal groups will be recognized by chromatin remodelers or reading enzymes with the chromodomains. And acetyl readers, they have bromodomains. So those would be the complexes that have bromodomains and would be recruited to those acetyl groups. And they can read the acetylation code. So maybe this figure explains better. So there is a dynamic interaction between all of those readers, writers, erasers, and readers. And this, of course, impacts different cellular processes. It's very important in maintaining cell homeostasis. It's very important for proper regulation of DNA templated processes, DNA transcription, DNA replication, DNA repair. It's very important to regulate cell differentiation. It's very important in stem cells, also in the induced pluripotent stem cells. It's important for the development, organ formation, and of course, when we have imbalance or deficiency in those chromatin remodeling events, this may be associated, this may lead to cancer, age-related diseases, and many other diseases. So this chromatin context and regulation of the gene expression within the chromatin context is critically important for the proper structure and function of our cells. And here we have more information and on the reader complexes, how they are recruited to the histone modifications, how they are able to read the histone code. So those reader complexes, maybe those big chromatin remodeling complexes that I've mentioned before that have multiple subunits, like this complex. So in this case, this is a ATP-dependent chromatin remodeling complex, and it's known that this complex would be recruited to the acetylated chromatin. So here, if we have acetylated histones, those are the marks for this complex. So this complex will be recruited, and then it can recruit additional complexes, and this will translate into a specific chromatin state. So here we have a process of spreading, so establishing specific chromatin and spreading of the chromatin states along chromosomes. So it all starts with the regulatory protein here in blue, and this regulatory protein is able to recruit a histone modifying enzyme. So this would be the writer protein. This could be a histone acetyl transferase. So this enzyme would acetylate histone here at this place, and this acetylation would be a signal for a different group of proteins for the reader. So the reader would be recruited here, and now the reader has the ability to recruit or interact with this writer protein. So by the interaction between those different readers, writers, and erasers, we can have the establishment of different chromatin states and also spreading of different chromatin states through long distances. I'll talk about that. Actually, I'm gonna stop here, so we'll come back to this slide and we'll finish about chromatin the next lecture.